a day before. Okay. So I already put the deadline for the submission the day before so that I can compile all the slides. Okay. Okay, any question? Boleh? Clear?
Clear.
Okay, so okay. Okay, can you see my slide?
Yes.
Okay. All right. So what I'm going to talk this afternoon is on genome mapping. Okay. Right. So bila you tahu map, you tahu lah map kan, macam kita nak memetakan. Okay. So it's actually you want to map the genome. Okay. So like I said tadi, genome is actually representative of the total repertoire of DNA. Okay. So in a particular organelle. Okay, and I think you know that plants have 3 genomes, okay, which are nuclear, mitochondrial, and chloroplast. Okay, with nuclear genome is the largest lah. Okay, and it's actually signify the vast majority of characteristic to an organism. Okay, right. So basically, genome mapping is actually approach to scan the genetic information of an organism for genes that are responsible for a specific trait. Okay, kita nak map the genetic information. Okay, so basically, higher plants are thought to have around 25,000 or more than 25,000 genes, and majority of the genes the function are yet unknown. Okay. So genome mapping will facilitate the isolation of the gene, okay, based on simply on the measurement of their effect on the phenotype. Okay, right? So a genome map can be thought of such as a roadmap, okay, the relative proximity of different landmark to one another. So dia samalah macam map kita ada landmark kan. Okay, and it's made possible by the fact that nuclear genome in higher organism is organized and transmitted as linear unit called chromosome. And DNA markers provide reference point that define specific places along each of the chromosome. Okay, so we want to define the location of the gene. Okay, so basically there are 3 categories of genome mapping approaches. One is genetic mapping, okay, which we're going to discuss in detail. Kedua is physical mapping, and keempat is actually comparative mapping. Ataupun kita dah buat hari itu, kita dah discuss comparative, which is we take the advantage of information of one species, for example, either physical or genetic, and kita akan deduce the probable gene arrangement in another species. Okay. So this is a comparative mapping, while physical mapping is to determine the proximity of DNA landmark by measurement of the physical quantity of DNA that lies between them. Okay, so this is physical mapping, while genetic mapping is based on recombination. You know, during meiosis, okay, there is process of recombination between chromosome. Okay, so it's actually the naturally occurring breaking and rejoining of chromosome to determine the relative proximity of DNA landmark to one another. Okay, based on the frequency at which they co-occur on the same chromosome segment. Okay, so this is genetic mapping. Okay. All right, so like I said, we're going to discuss further on genetic mapping. Okay, genetic mapping is also known as linkage mapping or meiotic mapping. Okay, where it refers to the determination of the relative position and distances between between markers along chromosome. Okay, so kita tengok markers, okay, because this marker will determine the location of the gene, okay, and so the genetic map distances between two markers are defined as the mean number of recombination events which involve a given chromatic in that region per meiosis. Okay, like I said, genetic mapping is based on the process of recombination and rejoining of chromosome during meiosis. So for construction of genetic map, okay, it requires researcher to develop mapping population, okay, decide the sample size and type of molecular markers for genotyping. So I'm going to discuss with you the different type of molecular markers. Okay, genotype the mapping population with sufficient number of markers and then perform linkage analysis. Okay, so then kita boleh map the distance between the markers along the chromosome. Okay, any questions so far?
No, no question.
Okay, so the construction of detailed genetic maps with high level of genome coverage is a first step to localize gene or to localize gene or quantitative trait loci that are associated with economically important traits. Okay, why we do genetic mapping? Okay, this is because we want to identify certain important traits dalam plant. Okay, this is part of dalam breeding program. This is what we want to select traits yang valuable. Okay, so we use for marker assisted selection, okay, to compare mapping between different species, okay, and also to establish framework to anchor a physical map, okay, and also the basis for map-based cloning of genes. Okay, so when we want to do genetic mapping, we use molecular markers. So the characteristic of molecular markers yang important, it should be highly reproducible, high throughput, co-dominant and transferable. Okay, so we want this marker, the characteristics of marker. Okay, so to construct detailed genetic maps with high level of genome coverage, bila you map the genetic, the genome, okay, you want to have coverage yang luas lah. Okay, it's the first step for some application of molecular markers in plant breeding. Okay, and these serve 5 purposes. First, the first purpose is to allow detailed genetic analysis of qualitative and quantitative trait. Okay, and also as part of the breeding program, you'll be able to facilitate integration of desirable genes. Maknanya you boleh combine genes yang you nak, valuable genes yang ni, okay, through marker assisted selection. Okay, and also allow comparative mapping between different species to evaluate similarity between genes, orders and function. Okay, and the fourth purpose is to provide a framework for anchoring this physical map based on chromosome translocation. And the final one is the purpose to constitute the first step towards positional or map-based cloning, okay, for economically important traits. Okay, so this is the principle of genetic mapping. Okay, like I said, genetic mapping is actually based on the recombination process of chromosome during meiosis. Okay, so it's based on the principle that genes, okay, or markers or loci segregate via chromosome recombination during meiosis. Okay, so which allow their analysis in the progeny. Okay, so you know during meiosis, chromosomes assort randomly into gametes, such that the segregation of alleles of one gene is independent of alleles of another gene. Okay, so basically Dr. Latif dah explain, this is stated in Mendel's second law and it is known as the law of independent assortment. Okay, And the law of independent assortment holds true for genes that are located on different chromosomes. Okay, but it does not always hold true for genes that are on the same chromosome. Okay, so when two genes are close together on the same chromosome, they do not assort independently. Okay, and you know they are said to be linked. Okay, and genes that are closer together or tightly linked will be transmitted together from the parent to progeny more frequently than those genes located far apart. Okay, so this is basically the process of meiosis lah. So at the beginning of meiosis, homologous chromosome will intertwine to form what we call as chiasma or chiasmata and exchange sections of the chromosome. Okay, so this process we call as recombination or we call as crossover or strand exchange, where then A molecules interact with one another to bring a rearrangement of the genetic information. Okay, so this is what we want to map lah. Okay, and the pair then breaks apart to form gametes with new combination of gene that differs from either of the parents. So this is genome mapping. Okay, so the observed recombinant fraction between two loci is actually estimate of 1/2 of the number of chiasmata or crossover events between two loci, because you know crossing over occurs at the 4-strand stage, and for single crossover events, only 2 of the 4 strands participate in the recombination. Okay, so basically I'm explaining the process of meiosis lah. Okay, if crossing over does not occur, the products are parental gametes. Okay, but if between the two types of gamete are the crossing over, the products are what we produce what we call as recombinant gametes lah. Okay, between two parents. Okay. Okay, so how can we decide how close two genes on one chromosome? Okay, so whereas genes located on different chromosomes assort independently, unlinked, and have a recombinant frequency of 50%, linked genes yang close together have a recombination frequency that is less than 50%. Okay, so the chance of crossover producing recombination between genes is directly related to the distance between two genes. Okay, so the lower the frequency of recombination between the two markers, the closer they are situated on the chromosome. Okay, conversely, the higher the frequency of recombination between two markers, okay, the further away they are situated on a chromosome. Okay, so based on this principle, we use molecular marker. Okay, so why kita construct genetic map, linkage map? Okay, construction, when we want to construct genetic linkage map, okay, it requires that we need to develop mapping population and decide the sample size. Okay, decide the type of molecular marker to be used, okay, for genotyping. Okay, screen parents for marker polymorphism because we want to compare between the parents. Okay, and the genotype, the mapping population. Okay, then kita map the genotype of the mapping population, which consists of the parents plus all the progenies. Okay, so that is mapping population. And then we perform the linkage analysis. Okay. Alright, so there are different type of markers, okay, yang can be used for mapping. Okay, so depends which one you want to use. Okay, so I will introduce you to different type of molecular markers. So the A markers are fragment of DNA indicating either mutation or variation, which can be used to detect polymorphism. Polymorphism ni kalau you use you know the word polymorphism, the differences between a loss of a gene for a particular sequence of DNA or different genotype. Okay, so such fragments of that marker are linked with a defined location within the genome, okay, and may be detected by using certain molecular technology. Okay, so there are different type of marker system. Okay, for example, you have RFLP, which is based on restriction fragment length polymorphism. Okay, we have RAPD, random amplified polymorphic DNA. We have microsatellites, or we call it a simple sequence repeats. We have AFLP and other type, the new generation of marker system, which includes SNP, which is single nucleotide polymorphism, CASPR, we have diversity arrays technology, and also genotyping by sequencing. Okay, so kita try tengok YouTube on RFLP. Okay, so this is one example of marker, okay, RFLP.
I think we should explore a few solutions.
Grammarly suggestions catch when you're
RFLP or restriction fragment length polymorphism is a non-PCR based marker technique to study genetic variation in individuals. RFLP is the first marker in the history of molecular markers. It exploits variation in the restriction site of DNA in different individuals. Now, what are restriction sites? Restriction sites are 4 to 6 base pair sequences which are recognized and
So for markers, you isolate DNA from the organism, from the plant. So you ada intact DNA, okay?
achieved by specific restriction enzymes. But what are restriction enzymes? Restriction enzymes, which is the basis of RFLP, are derived from bacterial defense system that cleaves DNA of invading viruses, thus protecting the bacteria. Such restriction enzymes acts as a tool to study DNA variations, which is essential if you want to know the similarities and differences between individuals or samples. The principle of RFLP is based upon restriction enzymes. Consider two individuals and their DNA. The restriction site of restriction enzyme is shown TTAA in the box. You can see the variation of a single base A, that is mutation in one of the bases of an individual. Thus the individual 1 has only one restriction site, but individual 2 has two restriction sites. When cleaved with a specific restriction enzyme that recognizes TTAA site, restriction fragments of DNA are generated. These restriction fragments are separated on agarose gel based on the fragment size. Smaller fragments move faster and travel longer distances towards the positive electrode. But at this stage, we cannot see the differences in the two individuals. The restriction fragments are then transferred to a nitrocellulose membrane and radiolabeled probes are applied. The probes are short DNA sequences which are complementary to specific DNA locus which is of interest. This is called southern blotting, in which DNA probes binds to their complementary DNA on the membrane. Here also differential pattern of the fragments are not visible. To visualize the different DNA fingerprint pattern between the individuals, X-ray film is applied to the blot and allowed to develop. X-ray film shows the restriction fragment pattern of two individuals. Now you can clearly distinguish two individuals based on their RFLP pattern. Now let's talk about the applications of RFLP markers.
Okay, so that is RFLP, okay, based on the concept of, all right, okay, RFLP, okay, you want to compare, you want to use molecular marker, okay, to compare between two individuals. Okay, so you isolate DNA from two individuals. Okay, then the DNA from two individuals being treated with restriction enzyme. Okay, so restriction enzymes are actually enzyme isolated from bacteria. Okay, which can cut DNA. Okay, macam tadi restriction enzyme tu kena dia recognize the sequence of TTAA, kalau tak salah saya. Okay, so bila dia the precision enzyme tu jumpa sequence of TTAA, dia akan cut the fragment of DNA. Okay, so then from there, okay, kita akan run agarose electrophoresis. Okay, then kita do Southern blot, kita label dengan radio label. Okay, dan kita boleh distinguish. So individu A tadi kita akan nampak dia akan radio label 1 10. Okay, kejap. Hello Siti, akak ada kelas? Tak tahu balik. Okay. Okay, alright. So this is RFLP. Okay, so RFLP based on the marker of based on dengan using restriction enzyme. So any question? Okay, you want to see another video? I think quite clear, kan? RFLP. Alright. And the second one is based on single sequence repeat based on microsatellite. Okay, so this is where this is a technique, molecular markers based on PCR. Okay, saya tunjuk you contoh.
Dapatkan kulit lembap berseri. Gabungan glutathione, hyaluron dan niacinamide. Dewy Radiance baharu. Berehat sebentar bersama KitKat.
SSR or simple sequence repeats. These are tandem or continuous repeats of 1 to 6 bases found in genomes of eukaryotes. SSR are also known as STR, short tandem repeats, microsatellites, SSLP. Based on repeat motif length, SSR may be mono, di, tri, tetra, penta, or hexa repeat. For example, AT is a di-repeat if it is found continuously in a single stretch for at least 6 times. For molecular studies, monorepeats are not well suited because of stuttering or smearing effect and poor resolution on gels. However, di-repeats are more suited because they show more variation than rest of the other repeats. Based on their occurrence in the genome, SSR may be genomic or genic. Genomic SSR may belong to coding, non-coding or intergenic region, but genic SSR are always found in coding regions. Genomic SSRs show more variability than genic SSR, while in terms of their amplification or transferability to related species, genic SSR are more cross-transferable. Now the question arises, why SSRs show variation? One of the main reasons for SSR variation is replication slippage. Because when DNA polymerase tries to replicate the repetitive DNA, it gets confused and it may lead to addition or deletion of one or more repeats. In this case, DNA polymerase finds it difficult to read 80 repeats and may increase or decrease SSR length in individual 2 and 3 respectively. Other reasons for the variation in SSR length are mutations and recombination during meiosis. Now we know that SSRs are prone to replication slippage. Individuals of a population have chances to show enormous SSR variation. Using SSR flanking primers, variation in SSR length is determined in different individuals. In this example, SSR marker is designed for 80 repeats. When this primer is used to amplify DNA of different individuals using PCR, similarities and differences between them is determined based on their length on agarose gel. In this agarose gel, individual 3 and 4 are having same length fragments, hence they are similar at DNA level than individual 1 and 2 who are different based on 5 base pair DNA led reference on the right. Advantages of SSRs as marker. SSRs are codominant, that is, they can distinguish between homozygous and heterozygous individuals. They are hypervariable, that is, they show high degree of polymorphism. SSRs are stable and the results are not influenced by the environment or developmental stage. Even 50 nanogram of DNA is sufficient for this study. SSR marker are sequence specific, that is, they amplify a specific DNA region. SSR markers can be automated using Sanger sequencing to study polymorphism. SSR assay is fast. Multiple markers can be checked in a single reaction and thus they are well suited for multiplexing. Their occurrence in the genome is random. SSR markers produce reproducible and reliable results.
Okay, so that is SSR marker. Okay, so the based on PCR. Okay, so you amplify using a primer. Okay, so you amplify the DNA from different individual. Okay, so you akan dapat different polymorphism. Okay, kalau the individuals are different. Okay, alright. So and we have RAPD also based on PCR amplification of random DNA fragment with primers of random nucleotide sequences. Okay, so it's quite similar to SSR. Okay, based on PCR juga. Okay, so polymorphism for RAPD are detected only by the presence of absence of a certain molecular weight. Okay, dia ada atau tak ada. Okay, dia akan between the individuals when you want to compare. Okay, you compare the presence of a or absence of a band. Okay, but RAPD ni dia non-reproducible and co-microation and it limit the application in the mapping. Ada orang tanya soalan ke?
Tak, doktor, dia tekan tadi.
Oh, okay.
Sorry,
Alright.
doktor.
Okay, so this is RAPD. Okay, then we have EST. Okay, I'll show you the video supaya you akan faham ni lah. Okay, so EST is actually express sequence tag. Okay, nama pun express sequence tag. Okay, maknanya apa yang being expressed. So this marker it will represent only the coding region of the genome because only those are the yang being expressed lah. So is the coding region. Okay, so EST marker. Okay, so I will give you a few papers to read. Okay, to help you to ni lah. Okay, untuk buat assignment. Okay, I will give you another video. Okay, so and another thing I want to inform you. Okay, if I'm not mistaken, kalau tak silap saya, kalau saya dah bagi assignment you markers, molecular markers ni, okay, saya tak ada soalan dalam exam.
Hello friends, welcome back to my channel. Today we will see a short topic on sequence tagged sites, and this comes under the short video series. If you are new to this channel, please press the subscribe button and click on the bell icon to get notified. So what are STS? STS, what's known as the sequence tagged sites, are 200 to 500 base pair unique short sequences in the genome. Its sequence and locations are known, and this can be very useful for genome mapping. So sequence tagged sites are detectable by PCR, and this can be done by designing primers. that can hybridize few base pairs apart from the region to amplify the STS region. So here you can see an example of sequence tag sites in the chromosome. So sequence tag sites that's derived from a region of DNA that is expressed, called as the expressed sequence tags. So these are the regions where the STS are expressed. For EST, for generating the EST, purified mRNA is used to generate the cDNA by reverse transcriptase. So the mRNA region is isolated and cDNA is generated by using reverse transcriptase enzyme by a process called reverse transcription for generating the ESTs. A map representing the order and spacing of the sequence tag sites within a stretch of DNA is known as the STS mapping. So a map can be generated by representing the order and spacing of the sequence tag sites within a stretch of DNA, and this is called as the STS mapping. To map STS or EST sites, a collection of chromosome fragments is examined for the presence of each STS or EST. And the fragments can be from single chromosome or from the whole genome. Hope you're clear with this short topic.
Okay, so for EST, nama pun express sequence tag. Okay, so kita akan isolate the RNA, okay, mRNA, kita convert to cDNA. Okay, then kita study the EST. Okay. Okay, so any question? Okay, so that's it. I have another topic which is gene expression. Okay, another two topic lah untuk gene expression and CMS. Boleh kan I request all of you to study first on gene expression minggu depan untuk lecture minggu depan. Pasal gene expression ni dia punya topik agak berat. Okay, and untuk CMS because topics untuk minggu depan pun ada yang akan masuk exam. So I don't want to rush. Nanti kan you tak faham sangat. I think is you overload code nanti. Okay? Alright? Boleh? Okay, so saya akan untuk gene expression saya dah masukkan dalam iTaklim. Okay, nanti kejap lagi by tonight I will upload all the papers related to our lecture. Okay, but I can assure you all the basic information untuk to be able to answer my exam question. Kita dah ni lah. Papers tu selalunya saya include untuk for your to understand better. Okay. So far kita dah cover 90% of the soalan exam yang nak masuk lah. Okay, alright. Any question?
Doktor,
Ya. Yeah.
kira pagi tu kelas, petang tu presentation
Ya. Yeah. Pagi tu kelas macam 10.
Depan tu last class ke doktor? Ke ada lagi?
So kena clear, saya tak akan ambil lain waktu. Saya akan pakai slot saya 10 hingga 12. But I hope I can finish before 12. Okay, then petang tu presentation. I think kita kena start pukul 2 juga. Sebab kita takut kita lambat. Okay? Okay, so
yang presentation tu online ke face to face?
Online.
Oh, okay doktor.
Online ya? Okay, so that's why I would like you to submit you punya slide a day before the presentation. Let me check bila saya suruh you submit. So that saya akan control slide you. Boleh?
Boleh doktor.
Okay. And kita dah set you punya time tu pun untuk you submit tu itu dah latest lah pasal saya kata by 19 hari bulan, kita dah kena keluarkan marks. Kalau tidak kita akan kena, kita orang akan kena ni lah, dapat reminder daripada office akademik. Jadi that's why that's the latest yang saya boleh ubah lah. Paling lewat you boleh submit you punya assignment. Don't disturb me. Koko. Kecil. Nak main tampal Koko. Main tampal. Kenapa? Jangan kecil. Okay, let me check. So you punya assignment untuk presentation, okay, I would like you to submit to me, okay, 16 Jun 2 hingga 5, presentation 16 Jun 2 hingga 5, but you have to submit to me on Wednesday, okay, by 2 PM. Kalau lewat sikit pun tak apalah, okay, tapi janganlah pagi tu baru you nak submit, jangan pagi hari Khamis because I want to arrange your punya presentation. Okay? So boleh tak you letak namakan file you tu based on your matric number, boleh ke?
Okey doktor. Boleh.
Alright, your matric number je, jangan letak apa-apa depan yang tu supaya saya nak susun ikut sequence. Boleh?
Okey doktor.
Okay. So saya letak ni Wednesday 15 Jun pukul 2 petang. Tapi bolehlah hantar pukul 9 malam pun boleh lah. So supaya saya just nak need time to arrange. Okay? Alright, so this presentation on plant molecular markers, you be tested on the content also lah dengan you punya presentation lah. Okay, so the rubric of the presentation, okay, you kena, saya ada letak rubrik okay, so kita pun ada kategori ni Q&A, okay, so kita akan ni lah bagi markah based on that also. Alright, okay, then video, I think you are quite creative, so I believe you can make video, interesting video on ni lah, assignment on video of sequencing, okay, then sequencing, okay, so you can submit to me which is the due date on Monday 13 Jun. Okay, sempat ke rasanya? Okay, you need more time. InsyaAllah sempat je. Kalau tak sempat saya extend.
Boleh juga doktor.
Okay, saya extend berapa bulan you nak. Tapi jangan dekat sangat lah because I can't afford, I want to mark kan. Can I give you until 16 hari bulan 6?
Okey.
Boleh doktor.
Okay,
Boleh,
saya
doktor.
ubah, nanti kejap lagi saya ubah 16 hari bulan 6. Maknanya you submit hari Khamis paling lewat 11.59 malam. Boleh
Boleh doktor.
Okay, thank you doktor.
Alright. Okay, no problem. So any other thing?
Video tu kena ada suara kita ke doktor?
Yelah kalau ada explanation. Tengoklah depends you punya creativity lah.
Okay boleh.
Kalau lagi menarik lah macam tadi tu dia explain kan. Alright. Right. So ini bukan first time you buat video kan? Bukan.
Ada. Dah pernah buat.
Okay. So, ni lah. Okay, saya akan extendkan sampai 16. Ini ada question related to soalan exam ke? Okay, saya ada calculation. So please bring ni lah. Apa ni? Calculator. Alright, any other thing? Any other question? Tak ada?
Tak ada.
Tak ada.
Okay, kalau tak ada, kita jumpa minggu depan. Okay, saya akan ambil slot saya je. Saya tak akan ambil yang lain lah. Boleh? Alright, so you baca gene expression dengan CMS. Okay, nanti saya by tonight, I upload the papers related to this molecular marker semua lah. Okay, dengan video semua I will give you the link. Alright, tak ada question. I'll see you next week lah ya.
Thank you, Doktor.
Betul.
You're welcome. But if you want information, let me know. Okay. Alright.
Okay.
Okay. Thank you.